Hey everyone, Dr. D here, and in this video, another interesting chapter from our Talaros Foundations in Microbiology 12th edition by Barry Chess. This is chapter 14.
Dr. D, 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 Dr. D. Explain stuff.
So, chapter 14, an introduction to host defenses and innate immunities. Let's go ahead and get started with this chapter. What are host defenses? They're talking about your immune system because you're the host to the pathogen, right? And look at this slide. There are two branches to the immune system that we're going to discuss. There's the innate immune system, also known as your natural immune system. And then there's the adaptive immune system, also known as the specific immunity, the specific immune system as well. This chapter is mainly about the innate branch of the immune system, and the next chapter is going to be more about the adaptive or specific branch of the immune system. I hope that makes sense. So, the innate immune system or natural immune system, which is the topic of this chapter, is present at birth. It provides non-specific resistance to infection. However, the adaptive immune system, which is the topic of the next chapter, this branch is specific. It must be acquired, okay? And it has a memory, and that's the big difference. So think of your innate or natural immune system as the branch of the immune system that you're born with. It doesn't improve over time. It doesn't have a memory. It doesn't remember an infection you had last year. It functions just as well today as it will probably next year, right? So the innate immune system, you can think of it as your basic defenses against infection, and you can think of the adaptive or specific immune system as one that forms a memory and one that actually improves throughout your life. Now, the immune system, again, you have either the innate branch or the acquired branch, but there are different lines of defenses. The first line of defense is part of your innate immune system, and it includes your physical and chemical barriers. Think of your skin, right? We're going to talk about it, but look here. We're talking about skin, tears, coughing, sneezing. All of these are your basic line of defense, right? What's actually keeping germs out of your system? That's your first line of defense. However, once germs break that through that layer of skin and get into your system, then it's the job of the second line of defense, which is also part of the innate immune system. And you can see that's where it involves other players in the immune system, including what are known as phagocytes, which we'll talk about, the inflammation pathway, fever, interferon, complement, right? We'll discuss all of these things. So again, your first two lines of defense against pathogens are part of the innate immune system. They function just as well today as they will tomorrow. They don't form a memory. They don't develop immunity, right? But they are protecting you. And so we're going to talk about these first two lines of defenses in this chapter. And then in the next chapter, we're going to discuss the third line of defense called acquired or specific immunity. And this one involves what are called the T cells or T lymphocytes and the B cells, B lymphocytes. And we're going to discuss that in the next chapter. So let's discuss your first line of defense, which is part of that innate immunity. We're going to discuss your physical or anatomical barriers at the body surface, mainly your skin, right? That's what we're talking about. Your chemical defenses, such as tears, saliva, earwax, and genetic resistance to infection as well. Your genes play a role in your susceptibility to various infections. Believe it or not, your skin is actually a pretty difficult place for microbes to grow. With these epithelial cells cemented together and impregnated with keratin, there's also a flushing effect of sweat. Sweat is salty and it flushes bacteria away. Your skin cells also slough off, right? So skin sloughs off all the time. So not all microbes are able to grow on the skin. You have to be well adapted to do so, such as the bacterium Staphylococcus aureus or Staphylococcus epidermidis. These are two species that are, more adapted to living on the skin. And that's why when you get a infection on the skin, oftentimes it's a staph infection. And that's because these microbes are actually well adapted to growing on the skin. And so if there's a compromise in that skin layer, well, then they're usually the first ones in there causing the infection. Isn't that neat to understand? You also have mucous membranes that are part of that first line of defense. In fact, mucus coats the digestive, genital urinary, and respiratory tracts. And obviously, your eyes have tears. The combination of tearing and blinking is also part of this first line of defense, keeping your eyes free of infection. Your mouth has saliva. Saliva is antimicrobial in and of itself because of various proteins inside of the saliva, which I'll talk about later. And also, you have the flushing effect of urination, defecation, vomiting. All of these can help keep you safe and are part of your first line of defense. In fact, everything you see here is part of that first line of defense. You have all of your surfaces, including the skin, but also the inside surfaces from your mouth all the way through the stomach, through the intestines, the colon, and exiting through the anus, right? All of that counts as your surfaces as well as inside of your nose. All of these are your surfaces, and all of them are kept safe because of their antimicrobial properties. The mucus on your body isn't there by accident. The mucus coat impedes attachment and entry of bacteria as well as viruses. So imagine you have all this mucus, right? Viruses, bacteria, they get stuck to the mucus and then they get flushed away. They can't penetrate through that mucus to get down to the skin layer to infect your skin. So mucus is doing a good job as one of your first line of defense at keeping you free of infection. Also, did you know the inside of your lungs have these ciliary cells? These cells that line the respiratory tree. You can see them here. These are epithelial cells of your trachea, your windpipe. And along your trachea, you have these ciliated cells. Remember how cilia work? Cilia are short beating hairs that do that power stroke motility. But you know what these cilia are for? Not for motility, but these cilia beat, and they beat debris out of your lungs and into the, digestive tract for destruction in the stomach acids. So when you breathe in dust, that dust lands on these cilia in your windpipe. These cilia beat and they beat in one direction, and that direction is bringing the debris back out of your lungs so that your lungs don't get infected. Isn't that neat? In fact, it's called the mucociliary blanket because it's a blanket of mucus and cilia, and that forms what's known as an escalator. All the debris is kind of moving towards the exit like an escalator, and then it goes down the esophagus into the stomach where those germs are destroyed. Other defenses in the tears, saliva, and skin include these enzymes called lysozyme. Lysozyme is an enzyme that hydrolyzes, remember hydrolyze means break, okay, the cell wall of bacteria. And yes, you have lysozyme inside of tears and saliva, so your saliva is actually antimicrobial. This lysozyme breaks apart bacterial cell walls. Isn't that neat? You also have defensins, which are peptides. Peptides are short proteins that lyse bacteria and fungi. You also have a high lactic acid and electrolyte concentration in sweat. This means that sweat is very salty. Not all bacteria can grow in salt, and it is also somewhat acidic. Not all bacteria can grow in acidic environments. So you see, the skin is actually quite a harsh environment, and only those microbes that are adapted can live on the skin. And then remember your stomach. Your stomach's actually very acidic. We're talking about a pH of 2 or lower. That's due to the hydrochloric acid in the stomach that not only does it help with digestion, but it also destroys any germs that you may ingest for the most part. There are also digestive juices and bile in the intestines. These have antimicrobial properties. The semen contains antimicrobial chemicals as well as the vagina, which has an acidic pH, which is maintained by the microbiota that live there that are producing that acidic environment. All of this is part of, again, your primary or first line of defense. All of these are helping keep you free of infection. And lastly, your genetics can play a role in your defenses, your innate defenses. If you have a mutation, that mutation may make you resistant to a particular pathogen. I'll give you an example. Humans carrying a gene for sickle cell anemia are naturally resistant to malaria. And so you can see how a gene edit or a gene mutation can result in immunity to a particular disease. Also with viruses, some people are genetically more predisposed to certain viruses than others, and some are more naturally resistant. So that leads us to our first concept check. Which of the following is not an example of the immune system's first line of defense? A, inflammation. No, see, inflammation occurs as part of our second line of defense. So this is part of our innate immune system, but it is not the first line of defense. It's technically the second line of defense. Lysozyme, that's an enzyme in our saliva, our tears, that's in our first line of defense. Hydrochloric acid, that's our first line of defense. The sneeze reflex, that's our first line of defense. The cilia in the respiratory tract, that is also our first line of defense. So again, the answer is A, inflammation, which is a part of our second line of defense. Still part of this chapter, still part of the innate immune system, but just not the primary or first line of defense. Now, the field of immunology really focuses on the second and third line of defense, the innate immune system and the adaptive immune system, what's going on inside of the body. And what is the primary function of the immune system? It's to surveil the body, looking for intruders, to recognize and differentiate normal versus foreign material, and then attack against and destroy the entities deemed to be foreign, right? So if a germ bypasses your first line of defense, it's your immune system's job to identify that and destroy it. And a big part of your immune system involves what are known as your white blood cells, or leukocytes, leukocytes is an all-encompassing term for white blood cells. And these white blood cells have an innate capacity to recognize and differentiate any foreign material. Remember, foreign material does not belong inside of your body. It's known as non-self. whereas your own proteins, your own cells, your own cellular structures are called self. Your immune system's job is to recognize self from non-self. Your immune system should not attack self, but your immune system should attack non-self. It should attack foreign material. So there are some really common foreign materials or non-self material, and these are known as pathogen-associated molecular patterns, or PAMPs. Again, these are molecules shared by microorganisms that are very common. For instance, peptidoglycan. Your white blood cells, the phagocytic white blood cells, which we'll get into in a minute, but we're talking about the macrophages, the dendritic cells, and the neutrophils. Those white blood cells have receptors called pathogen recognition receptors, or PRRs for short, and these receptors recognize those PAMPs. They recognize patterns on foreign materials such as peptidoglycan, flagellin, the protein that makes up the flagella, LPS, which is the component on the outside of gram-negative cell walls, right? Those are common patterns that are recognized by your white blood cell receptors. So when you think of your phagocytic white blood cells, know that they have these receptors called PRRs, and those receptors recognize these common patterns called PAMPs. Now, your body compartments that participate in immune function include the reticuloendothelial system, or RES, the extracellular fluid, or ECF, the bloodstream, and your lymphatic system. And fluid is shuttled from one of these compartments to another. So you see, blood can leave the blood vessels. Blood can enter the ECF. The ECF can enter the lymphatics. The lymphatics can leave and enter the blood. So fluid is constantly crossing from these different bodies of fluid. And when they do so, those are opportunities for non-self foreign matter to be recognized by white blood cells. Because look right here, white blood cells exist in the ECF. White blood cells are always hanging out. And these white blood cells are just screening your fluids for non-self matter, like those PAMPs we just discussed. A little bit more about the reticuloendothelial system, or RES. It's a network of connective tissue fibers that interconnects other cells and meshes with the connective tissue network surrounding organs. So you can think of it as tissue and fibers that surround organs. And these tissues are full of phagocytic cells. Remember, these are the white blood cells that phagocytose or engulf foreign pathogens. Remember, these are the cells that have those receptors, the pattern recognition receptors, and they recognize those common patterns called PAMPs. And these phagocytic cells make up the mononuclear phagocyte system, macrophages ready to attack and ingest microbes that pass the first line of defense. So for instance, if you breathe in, you might breathe in some microbes, right? Those microbes will find their way into your lung. And if those microbes try to bore their way through the lung, on the other side, they'll find these macrophages. These are going to be the alveolar macrophages or lung macrophages, ready to gobble them up the second they try to enter your body through the lung. So again, you see here, these are your cells, your tissue cells. And in this network of fibers between your tissue cells are those white blood cells, those phagocytic white blood cells like neutrophils depicted here, dendritic cells depicted here, and macrophage cells depicted here. They're just waiting in this RES, waiting for a pathogen to try to enter your body. They will then recognize that pathogen by recognizing its PAMPs with its PRRs, and then they will phagocytose that invader before it can, make a home inside of your body. Now let's talk about the blood a little bit more. Whole blood includes plasma and formed elements. These are the blood cells. When we're talking about the serum, it refers to the liquid portion of blood after a clot forms, minus your clotting factors. So here you can see the serum on the right. This is the liquid portion of blood after a clot forms. The clot is shown here at the bottom, and the whole thing is whole blood. This is whole blood. Plasma, when we refer to plasma, we're referring to the 92% water, but this plasma includes metabolic proteins, globulins, clotting factors, hormones, and other chemicals and gases to support normal physiological function. So here you can see this is unclotted whole blood. This layer at the top is called plasma, and underneath that is called the red blood cells. So this is what your blood looks like outside of the body. Now, something really neat is that your blood cells all come from stem cells that live in the bone marrow, the red bone marrow. So your bone marrow possesses these stem cells, and these are known as hematopoietic stem cells. And these stem cells are amazing. They can differentiate, that means change, into all the different types of blood cells in your body. Did you know in your body you have platelets that are formed from hematopoietic stem cells, also known as thrombocytes, red blood cells called erythrocytes, and then the leukocytes, remember the white blood cells. And there are many different white blood cells, but they all originated from one original cell, right? Those cells are called the hematopoietic stem cells. So your bone marrow is so important because it houses these stem cells, and these stem cells are what generate your blood supply. A red blood cell doesn't divide to give you two red blood cells. In fact, it can't. Red blood cells don't even have a nucleus, right? So where do red blood cells come from? They come from hematopoiesis, the process of making blood with those hematopoietic stem cells. And remember when I said there are many different white blood cells? Well, they're broadly categorized as either granulocyte white blood cells or agranulocyte white blood cells. So we're going to talk about both. The granulocytes have a lobed nucleus, while the agranulocytes have an unlobed, rounded nucleus. So let me tell you about the different types of blood cells. So this is a nice overview of how hematopoiesis occurs. Remember, hematopoiesis is the process of differentiating into the different types of blood from those stem cells, the hematopoietic stem cells in the bone marrow. So let's zoom in to take a look. Again, at the top of this tree is the hematopoietic stem cell, the stem cell that resides in the red bone marrow. This stem cell can pick one of two pathways. It can either pick the myeloid pathway and become these cells over here on the left, or it could pick the lymphoid pathway and become one of the cells down here at the bottom right. Let's look at the left side first. Remember, the hematopoietic stem cell can give rise to erythrocytes, which carry oxygen and CO2 in your body. They can become the platelets which are involved in blood clotting and inflammation. And remember, these two cells are not considered white blood cells. These are not white blood cells. The rest of these cells are considered white blood cells or leukocytes. So first, the granular leukocytes. These are the ones with the multi-lobed nucleus, right? The lobed nuclei. Notice how the nuclei looks irregular and lobed. These include the neutrophils, eosinophils, basophils, and the mast cells. Those are the granulocytes. Next, the agranulocytes or agranular leukocytes. This includes the T cells, or also known as T lymphocytes, the B cells, or B lymphocytes, which give rise to plasma cells, and the natural killer cells, or NK cells, as well as the monocytes, which give rise to the macrophages and dendritic cells. So that's a mouthful. There's a lot of different players here. I bet you didn't know there were that many different white blood cells, but we're going to discuss each and tell you what each of these does. Let's start with the granulocytes, these granular leukocytes. Let's start with the neutrophils, eosinophils, and basophils. Again, notice how they have that lobed nucleus, but also notice how all of these granulocytes, the way they got their name is that they have these little granules inside. It almost looks like salt and pepper. You've got like pepper inside. Those are the granules. And those granules possess reactive molecules that can be released, right? So think of these as little molecules, little granules full of molecules that can be released from the cell. Okay, so again, let's start with the neutrophils, eosinophils, and basophils. The neutrophils are phagocytes. You should remember that neutrophils are big-time phagocytic white blood cells. Their job is to identify foreign microbes with their PRRs, remember the pattern recognition receptors, and then phagocytose them, bring them in as food, right? Like engulf them as food. Now, the eosinophils are really neat. They destroy eukaryotic pathogens. So think about protozoa, think about worms, right? Helminths, that's their job, to attack those. And by the way, eosinophils are minor phagocytes as well. Next, the basophils. The basophils they use their little granules, to release potent chemical mediators, such as histamine. And then lastly, let's talk about the last granulocyte, right? We talked about neutrophils, eosinophils, basophils. Let's talk about the mast cells as well. The mast cells also release histamine. Both the mast cells and the basophils are able to release histamine. You may have heard of histamine, right? Histamine is released during inflammation, but it is also released on accident, during allergies, right? So a lot of allergy medications are antihistamines. Here's an up-close picture of a neutrophil. Notice the multi-lobed nucleus. Here's an eosinophil. Usually these have a two-lobed nucleus. Here's the basophil. Now let's discuss a little bit about the agranulocytes. These ones here, the agranular leukocytes. So let's discuss these, the monocytes, macrophages, dendritic cells, B cells, T cells, and the natural killer cells. Let's talk about these. These have a proper nucleus. Notice how the nucleus is round like a normal nucleus. All right, let's talk about the B lymphocytes and the T lymphocytes first. The B lymphocytes are known as your B cells. They partake in the adaptive or specific immune system, a process known as humoral immunity. I'll explain that in the next chapter. And when these B cells become activated, they can produce antibodies. Have you ever heard of antibodies, right? Antibodies are produced by activated B cells. In fact, those activated B cells turn into what are called plasma cells, and it's the plasma cells that are antibody factories. The T cells, or the T lymphocytes, these partake in what's known as cell-mediated immunity, something I'll explain in the next chapter. And these T cells are responsible for destroying any of your cells that happen to be infected with foreign material. We'll talk way more about how the B cells and the T cells work in the next chapter. Now, one thing you should remember is the monocytes become the macrophages and dendritic cells. Let me show you what I'm talking about. Do you remember I said that the monocytes can differentiate, that means change into, the macrophages or the dendritic cells? In fact, these monocytes, they don't linger in the body long, only about 8 hours. After about 8 hours, they pick a fate. They either become a macrophage and live in a tissue, or a dendritic cell living in a tissue. And another thing you should know is that monocytes, macrophages, and dendritic cells are important phagocytes. They're phagocytic white blood cells. They have those PRRs that recognize those patterns, those PAMPs. Here's a close-up picture of lymphocytes, monocytes. Next, let's talk about the lymphatic system, an entire system of vessels and nodes that allows this fluid to pass through it, and that fluid is extracellular fluid. We've all heard of the cardiovascular system, right? Veins and arteries for transport of blood. Well, there's an entire other set of vessels that are transporting not blood, but lymph fluid. And that lymph fluid is known as extracellular fluid. And that fluid may have foreign matter in it. Let's say you have a viral infection or a bacterial infection. That fluid may have that foreign matter in it. So it's a great place for white blood cells to filter this extracellular fluid, searching for that foreign matter, right? Foreign materials, foreign proteins, foreign sugars, PAMPs, right? Viruses, anything foreign and non-self. So think of the lymphatic system as a meeting place for white blood cells and foreign pathogens. And here is a drawing of the lymphatic system. It's depicted here as these green vessels. And notice how these green vessels meet up at nodes. Have you ever heard of lymph nodes? Well, that's what we're talking about here. Your lymph nodes. You have several lymph nodes in the inguinal part of the body. You have several near the armpit. You also have several in the neck, right? When you go to the doctor and the doctor feels below your jaw here in your neck region, the doctor is feeling for those lymph nodes, because those lymph nodes become swollen when you have an active infection, right? And that's because those white blood cells are proliferating, and those white blood cells are attacking foreign substances in your body. That's why those lymph nodes become swollen up. But now you know how lymph nodes work. Lymph vessels feed extracellular fluid to lymph nodes, and those lymph nodes are actually full of B cells and T cells and immune system cells. And that's where foreign matter meets up, says hi, with those white blood cells that are just waiting to meet them. By the way, an important and interesting fact about the lymph system is that the fluid only moves towards the heart. It's not like the cardiovascular system. You guys familiar with the cardiovascular system? Blood flows away from the heart through arteries and back to the heart through veins, right? You know that. But the lymphatic system only goes one way, towards the heart, towards the heart. And again, the lymphatics are full of this fluid called lymph fluid, which is a plasma-like liquid carried by lymphatic circulation. That's known as your extracellular fluid, essentially. And lymph is formed when blood components move out of the blood vessels into extracellular spaces, the extracellular fluid. This lymph is made up of water, dissolved salts, and some proteins. It can transport white blood cells, fats, cellular debris, again, which may or may not contain foreign matter and infectious agents as well. You might be wondering how lymph moves. I told you those lymphatic capillaries gather up that extracellular fluid, and then that fluid makes its way to my heart, right? The fluid moves through those vessels towards my heart. Well, did you know that that fluid moves because of my muscles contracting? So every time I flex my muscles and I move my hands and I flex my hands and I do things or if I'm walking, through skeletal muscle contraction, right? The contraction of my skeletal muscle, just my muscles, that's what's pushing the lymph fluid towards my heart. Isn't that neat? So again, in the cardiovascular system, arteries carry blood away from the heart, while veins carry blood towards the heart. Those lymphatic ducts and vessels, the lymphatics only move fluid towards the heart. And what ends up happening is that lymph fluid dumps into a vein near the heart. So basically, this lymphatic trunk collects the fluid and it kind of dumps it into a vein near the heart. So fluid can leave the lymphatics and enter the blood. It can leave the blood and enter the lymphatics. And that fluid, it can move back and forth between the two systems. And again, these lymph nodes are just nice little meeting spaces between white blood cells and that extracellular fluid, which may contain foreign matter. And that's a great place for white blood cells to identify foreign material. All right, so we're about at the halfway mark here, so you know what that means. We got to take our break time with Gizmo and Wicket, see what these little guys are up to, and we'll be right back with more. Now let's talk about lymphoid organs and tissues. These are important organs and tissues for the immune system. There are primary organs. These include the thymus and the bone marrow. These are sites of lymphocytic origin and maturation. That means this is either where lymphocytes come from or where they mature. So I'm going to tell you this now, but I'm going to, obviously I'm going to talk about it in more detail in the next chapter. But you should know that, again, the bone marrow is so important. It contains those hematopoietic stem cells that give rise and can differentiate into all the different types of blood. And one thing you should know is that not only does the bone marrow create the B cells or B lymphocytes, but you should make a note that that's also where the B cells mature. B cells mature in the bone marrow. Now, one thing that's good about the thymus that you should know is that the T cells or T lymphocytes mature in the thymus. And we'll talk way more about what that means to mature, right? But for now, note that the B cells mature in the bone marrow, while the T cells mature in the thymus gland. So that leads us to the secondary organs and tissues of the lymphoid system. And these are great places for that extracellular fluid to meet up with microbes, okay? These include those lymph nodes I told you about, the spleen, which just teeming with B cells and T cells, the MALT, the GALT, and the SALT. Okay, what does that refer to? MALT refers to the mucosal-associated lymphoid tissue. GALT refers to gut-associated lymphoid tissue. And SALT is skin-associated lymphoid tissue. In fact, you could see them here on this image. You can see you have GALT in the gut. You have MALT in the mucous membranes. Okay, so that's it for the secondary organ tissues. Let me tell you a little bit more about the thymus. The thymus is a really neat little organ. It's a butterfly-shaped organ in the upper chest area. it actually starts shrinking after puberty. So it has a high growth and activity until puberty, and then it actually begins to shrink, and it pretty much becomes completely depleted by the time you're elderly. And remember what I wanted you to remember about the thymus? It's the site of T cell maturation. And remember the lymph nodes? We already talked about those lymph nodes. Those lymphatic vessels feed into the lymph nodes, small encapsulated bean-shaped organs along lymphatic channels and large blood vessels of the thoracic and abdominal cavities. And remember this, the job of the lymph node, first of all, it houses tons of B cells and T cells. It filters that lymph fluid and provides niches for immune reactions. When those immune reactions happen because you have an infection, that's when those lymph nodes swell up. And that's exactly what your doctor is feeling for when they feel under your armpit, or they're feeling in your throat or in the inguinal region. They're feeling for those inflamed lymph nodes, a sure sign of infection. And nestled below the diaphragm and just left of the stomach is the spleen. The spleen is somewhat like a lymph node. It is full of those white blood cells, B cells, T cells. The spleen filters circulating blood to remove worn out red blood cells. But basically, the spleen has two main jobs. One is to filter out the old red blood cells, but the other job is to serve as a meeting place for white blood cells and foreign matter in the blood. All right, concept check number 3. Are you ready for this? Where do T cells mature? I told you to remember, that was the thymus. The thymus is the answer there. Now let's talk about another part of the second line defenses called inflammation. Inflammation is very important as a second line defense mechanism. One thing you should realize is that any traumatic event, any kind of tissue damage, can result in inflammation. Even if I poke my skin with a sterile needle, that tissue damage alone will trigger inflammation, right? So inflammation does not have to be caused by pathogens. Tissue damage results in inflammation. And these are the signs and symptoms of inflammation. You should know that anytime you have inflammation, there's redness, also known as rubor. You should know these terms as well. Redness or rubor. The redness in the inflamed part of your body. So like, look at this. This is an inflamed fingernail here. This is an inflamed, like it looks like an ingrown fingernail here. When you have inflammation, you have redness, also known as rubor, increased circulation, right? You've got blood flow, and vasodilation. Vasodilation means dilated blood vessels, right? So that's what happens when you get inflammation. The blood vessels dilate. That's what vasodilation means. And if the blood vessels are dilated, doesn't that mean there's more circulation, more blood flow to that region? And wouldn't that cause redness? Because think about it. Bigger blood vessels, more blood, it's going to turn red, right? Next, because all that warm blood is rushing to your finger, right? It's going to feel warm. This is known as calor. Warmth is calor. Heat given off by the increased blood flow. Because remember, blood is warm. And then all that extra blood flowing to that area is going to cause swelling, also known as tumor. Increased fluid in the tissue as blood vessels dilate. This is forming an edema. White blood cells, microbes, debris, and fluid collect to form pus. Pus forms, and it prevents the spread of infection. And isn't this whole thing painful, right? So pain is obviously associated with inflammation as well. This is known as dolor. This is the stimulation of nerve endings due to all that swelling. Here's what happens when you have tissue damage. Remember, so here, look, it looks like some sort of nail has punctured the skin. Up here is the skin. Here is the extracellular fluid, and that's a blood vessel down here. These round things are mast cells. Mast cells. What do mast cells release? Can you beat Wicket? Do you remember what are the mast white blood cells good for? That's right, Wicket. They release histamine. Okay, so let me explain something real quick here. The first thing that happens when you have tissue injury is that the very, very first response is vasoconstriction. The blood vessels constrict. The blood vessels constrict because what your body doesn't want is, if you have sudden trauma, your body doesn't want you to just bleed out, right? So those blood vessels constrict at first to prevent too much loss of blood. Does that make sense? But at that point, the mast cells release their histamine, and that histamine causes what's called vasodilation. So next, the blood vessels dilate. There's vasodilation. And that's what leads to that increased blood flow to the area. And remember, the swelling and the pain as well. At this point, you're going to have white blood cells such as neutrophils there, phagocytosing any bacteria or any foreign pathogen. This will lead to edema, swelling, and ultimately the formation of pus. And those neutrophils are a main component of pus. And then finally, this will result hopefully in resolution and scar formation. And this is where everything is being repaired. Macrophage lymphocytes and fibroblasts migrate in, initiate immune response, and repair of injury. This is an interesting image of a blood clot. After an injury, fibrinogen is converted to insoluble fibrin, and that forms all these little threads you see here. These threads ensnare red blood cells, and then the white blood cells and platelets are also trapped in this network, right? And that helps to stop bleeding. Isn't that neat? So this is an actual image of clotting with an electron microscope. Another thing that happens here during inflammation is not only do the blood vessels dilate, but these skin cells, the damaged skin cells, release signals called cytokines. And think of those signals as kind of an SOS, like help me, help me. And those are called a chemoattractant. Chemoattractants are chemicals that bring other cells to the site. And when there's vasodilation and release of these chemokines, neutrophils start migrating to the site of the injury. By the way, the histamine also opens up leaky spots in the blood vessel. Let me show you what that looks like. See here, this is a blood vessel, and these are the leaky spots that open up in the endothelial cells, right? Endothelial cells are the blood vessel cells. They actually open up little pores. And do you remember I said that the damaged tissue releases those chemotactic factors, those factors that say, help me. And so what happens is something really neat. The neutrophils in the blood stick to the blood vessel, and that's called margination. Next, the neutrophil, remember, which is a phagocytic white blood cell, it actually contorts itself and squeezes. Look, it squeezes through those little openings in the blood vessel. And this is a process known as diapedesis or extravasation in some textbooks. And that allows the neutrophil to actually get to the site of tissue damage, phagocytose any bacteria, form pus, resolve the disease, so that repair can occur. Isn't that neat? Now, something that happens to you when you have an infection is called fever. Fever is actually an adjunct to inflammation. They usually go hand in hand. Fever is triggered or initiated. All right, you guys, Punky's come to say hi. Hi, Punky. When she's purring, I don't know if the mic picks up the purr. Can you hear that? Punky? She's got such a sweet purr. Fever, when your body has a fever, it's because your own cells have triggered the fever due to usually an infection, right? So what happens is your own cells release these molecules called pyrogens. Pyro meaning fire, okay? And that's because your body feels like it's on fire once, you have a fever. But those pyrogens, oh, she's trying to drink my coffee here. That's not for you, little one. But those pyrogens, what they do, your cells release these signals called pyrogens. Those pyrogens travel to your brain, to the hypothalamus in your brain. And you can think of the hypothalamus as your body's thermostat. Just like when you're at your house and you want it to be warmer, you can go turn up the thermostat. That's what the pyrogens do. Your own, let's say, macrophages, remember your phagocytic macrophages? They release pyrogens. The pyrogens travel up to the brain and reset your brain. They tell your hypothalamus, hey, increase the body temperature. And then your body responds by increasing the temperature. Isn't that neat? It signals muscles to increase heat production and vasoconstriction. Remember, vasoconstriction refers to blood vessel constriction. Isn't that neat? So again, remember, usually these pyrogens are released by your own white blood cells like monocytes, neutrophils, macrophages during phagocytosis. So when you have an infection. But do you guys remember when I told you about, oh, is she going to jump? Okay. I don't know what she's doing, you guys. Oh, yep, she jumped. She's going to bed. She's so sweet, you guys. But there is also another way to get it in a fever, and that's called exogenous pyrogens. So for instance, do you guys remember I told you about endotoxin? What was endotoxin? Do you remember? I told you to remember this. What is endotoxin? What is another name for endotoxin? That's right, Wicket. Lipid A. Do you guys remember that the outer part of the outer membrane of gram-negative cells includes LPS, and the lipid portion of LPS is called lipid A? Well, lipid A is a synonym for endotoxin. It's the same thing, okay? So if you have a ton of this lipid A or endotoxin in your system, that's going to trigger a fever, right? Or if you don't have lipid A, it's your own monocytes, neutrophils, or macrophages that can release these pyrogens to reset your hypothalamus, triggering a fever. So it's normally your own body that triggers a fever. And so think about it. If your own body, if your own macrophages are trying to trigger a fever, then there must be some kind of benefit to fever. And that's true. There are many benefits to fever. Fever inhibits multiplication of temperature-sensitive microbes. It also impedes the nutrition of bacteria by reducing the available iron in the blood. Bacteria need a steady source of iron in order to grow. And having a fever causes what's known as hypophoремia, which means low iron in the blood. And not only that, but fevers increase your metabolism and stimulate your immune cells. They stimulate immune reactions and protective physiological processes as well. Right, Punky? And remember, when those phagocytes, those white blood cells are moving around your body, their job is to survey tissue compartments and discover any microbes that might be hiding, any particulate matter, and dead or injured cells. They then ingest and eliminate those materials, and they can even extract immunogenic information from foreign matter. This is going to be discussed in the next chapter. We're gonna talk about the step 3 in in the next chapter when we talk about the adaptive or specific immune system. Again, do you guys remember all the phagocytic cells? Neutrophils are phagocytic, macrophages and dendritic cells are phagocytic, and even, I didn't mention this before, but B cells are phagocytic. And do you recall what I mentioned before? And that is that eosinophils have a minor phagocytic role as well. And do you remember how phagocytosis works? We talked about it a little bit earlier, but this is a step-by-step. This is a step-by-step where it's showing you all the steps in phagocytosis and how that works. So let's break this down together. All right, first of all, take a look here. First of all, the phagocytic cell has to migrate to the, oh boy, she's trying to attack the mouse. Punky's trying to attack the mouse, making it hard to see. So first of all, the phagocyte has to move to the site of the infection, right? That's called chemotaxis. And you see this often with neutrophils. Remember I showed you during inflammation, those neutrophils move to the site of the infection. Next, once the phagocyte is in the vicinity of bacteria, do you remember that bacteria have structures on their surface that are common pathogen-associated molecular patterns called PAMPs? Well, look at this. The macrophage has receptors called pattern recognition receptors, and those receptors recognize common PAMPs on the surface of bacteria and other microorganisms. Remember, like for instance, peptidoglycan is a PAMP, and that binds, right? So the bacteria just bound to the PRR receptor on the phagocytic white blood cell. This triggers what? It triggers phagocytosis. See, the membrane invaginates and brings those bacteria in, right? And it forms a phagocytic vacuole. Look at this. This phagocytic vacuole contains the bacteria that was just ingested into the cell. These are the bacteria in blue. This is a membrane surrounding them. And all of this whole structure is called the phagosome. And do you remember at this point, we want to destroy these bacteria, remember? So what we do is we take a lysosome. Do you guys remember that lysosomes are the digestive organelle of a eukaryotic cell? You see these green dots? These green dots are hydrolytic enzymes. These are enzymes that digest organic material. By the way, it's so hard to concentrate because she keeps attacking my mouse. I wish you guys could see this. Okay, I got her. So then you remember what happens? The phagosome full of bacteria fuses with the lysosome full of hydrolytic enzymes. And when this happens, it forms what's called the phagolysosome. Those enzymes chew up the bacteria because those enzymes are, designed to chew up organic material, and that leads to the destruction of the bacterial cells. Okay, and once those bacterial cells are destroyed, then you have cellular debris, right? This is called cellular debris. And that debris can be released from the cell through exocytosis. Isn't that interesting? So this all is the process. These are the sequential events in phagocytosis. Next, let's talk about the interferon pathway, an important process in that innate immune system response, the second line of defense. In this interferon pathway, small proteins are produced by certain cells in response to a viral infection. This happens when a cell becomes infected with a virus. It can produce interferon. There are three major types of interferons, but we're not going to talk about the different types here. All of these are produced in response to viruses, RNA, immune products, and antigens. So here you can see the antiviral activity of interferon. Here's what happens. Punky changed the slide. I'm sorry. So here's what happens during the antiviral activity of interferon. First of all, a cell, you see the cell on the left? The cell becomes infected with virus. Look at this. The cell becomes infected with virus. And you guys know what that means. Once a cell in your body is infected with virus, then it becomes a viral assembly factory, right? The virus copies its DNA or RNA, it copies all its proteins, and then assembly and release, right? Well, under certain conditions, this can trigger the cell to turn on the interferon gene and turn on the interferon pathway. The interferon gene gets triggered when a cell realizes that it's infected with virus. The interferon gene codes for an interferon protein called IFN. Then interferon protein, shown here in orange, is secreted from the cell. It's released from the cell. That interferon protein travels to an adjacent cell, so like a nearby cell, and it serves as what's called a cytokine or a signal. And that signal, see, the signal has a receptor on the adjacent cell. It's called the interferon receptor. See in purple? In purple is the interferon receptor. And when the interferon binds to the interferon receptor on the adjacent cell, this signals to the cell to activate certain genes. What type of genes, you may ask? Genes that code for antiviral proteins. These are proteins that will actually destroy viral nucleic acids, viral DNA, viral RNA. So check it out. At this point, look what's going to happen. This cell on the right has a heads up, right? It has a heads up. It says, its neighbor said, help, his neighbor said, it's too late for me, but it's not too late for you, right? And so this cell had a heads up, and he was able to prepare by producing these proteins that degrade nucleic acids, right? Incoming nucleic acids. So unfortunately, interferon did not help this guy on the left. This guy on the left is pretty much toast, but his neighbor is going to be okay because the neighbor got the heads up. The neighbor produced those antiviral proteins. So what happens is the neighbor can still get infected with virus, but the virus cannot take over that cell because the minute the uncoding happens and the viral nucleic acid is in the cytoplasm, it gets degraded and chewed up, right? So what you should know is that the interferon pathway is a response to viral infection. It involves this interferon pathway, and it doesn't prevent the adjacent cell, the neighboring cell, from being infected, but it does prevent the neighboring cell from being taken over. So make a note of all of that. All right, and then the last component of the innate immune system that I wanted to talk about, again, part of the second line of defense, is called complement. Complement is really interesting. complement is part of your immune system, but it's made up of only proteins, serum proteins. That means proteins that float around in your blood serum. These proteins play an active role in your immune system. The way that these proteins get activated, because when you don't have an infection, these complement proteins are inactive. But when you do have an infection, these complement proteins become activated. And do you see here, these are the three ways by which complement proteins become activated. There's the classical pathway, the lectin pathway, and the alternative pathway. I don't need you to remember any of this, but I just wanted to leave it here so that you know that these proteins become activated. And these complement proteins, there's about 30 of them. And each one has a different role, or they can work together in your immune response, right? As part of your immune system. And these complement proteins, their names start with C. So take a look at this. Complement protein C5b, C6, C7, C8, and a bunch of C9s embed themselves into bacterial membranes and cause the membrane to fall apart. And this structure that forms is called the membrane attack complex. Isn't that neat? So once these complement proteins get activated, these ones embed themselves into the membrane of bacteria, forming a MAC or membrane attack complex, and that causes the cell to lyse. Isn't that neat? So I just showed you the membrane attack complex or MAC. It uses activated complement protein 5b, complement protein 6, complement protein 7, complement protein 8, and a bunch of complement protein 9s. There's another complement protein called C3b, and this one plays a role in opsonization, which means it sticks to bacteria and flags them for phagocytosis. And then there's another complement protein that once activated, this one's called C5a. Once this one is activated, it promotes chemotaxis of neutrophils, which means it says, hey, neutrophils, come here, right? It's a chemotactic signal. It's an attractant for neutrophils. And by the way, C3A, I should say, C3A and C5A also promote vasodilation. Isn't that neat? So these complement proteins have actual roles in your immune system. Once they get activated due to an infection, they can actually play a big role in your defenses. Now that leads to the final concept check of the chapter. And even Punky is feeling exhausted, so we need to wrap this up. Which of the following defenses is most likely to be active during a viral infection? Inflammation, fever, interferon, and complement. Can you beat Wicket? Let's see if you could beat Wicket to the punch on this one. That's right, Wicket. Interferon, right? Because that's a big role of interferon pathway, is to protect your cells from the spread of viruses. So that's it. I believe we've reached the end of this really interesting chapter. And the next chapter is even more interesting. The next chapter is all about B cells and T cells. And that will tell you about how your immune system really works and forms a memory. Have you ever heard, about your immune system? You can become immune to certain things, or how vaccines work. Vaccines make you immune to different bugs. How does that work? That's all due to memory. And that all has to do with the next chapter. So I'll see you there.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.